In the search module, you can query either a cell signal molecules or target genes. So there are two usages. The first usage of the search module is querying target genes of a cytokine. For example, you can input BMP6 in the search box and click search. Then you will get a selection interface like this. The cytokine database only included cytokines with data curated from public repositories. Thus, the result may not be available sometimes for understudied cytokines. There are two parameters on this page. The first parameter is the maximum number of entries to display ordered by the adjusted value. Each cytokine treatment profile may contain log4 change values for about 20,000 human genes. Also, a cytokine may have many treatment profiles from various studies and cell models. Thus, enumerating all results on the web browser will take a significant amount of time. The system will rank all target genes by their absolute log4 change values and only show top results. The second parameter is the selection between signaling molecules or target genes. For a cytokine name, they could either serve as a signaling molecule regulating many targets or target genes with upstream cytokine regulators. Such ambiguity will not exist if the input is only a target gene, for example, SOX4. For the BMP6 example, we will select the first option and click Submit. The searching result may take a little while, since the system will go across many MySQL records. The search results are shown in a table with multifunctional headers. Mouse over each column name will trigger the explanation of each field. Mouse click on each column will sort rows in either ascending or descending order. The row selector will pop up once you put the mouse cursor on the space beneath the header. You may input a keyword to select a row, a set of rows using the column value. For example, you can input true in the search box beneath the column high quality. To only analyze treatment profiles that passed our quality controls, the row selector will affect the analysis modules introduced later. The meaning of each column is available below. The model means the cell model used in the cytokine treatment experiment. The duration means the duration of experiment treatment, if available in our annotation. The dose column means the dose of treatment experiment, if available in our annotation. The dataset means the database ID for the data in each row. Platform means the transcriptomic profiling platform, such as microarray or RNA-seq. Details presents further detail of the platform. Gene means the target gene name. Value presents the log2 fold change between treatment and control conditions. Value adjusted presents the adjusted value through normalizing the original log fold change by the standard deviation across all values in each treatment profile. The high quality column indicates whether the current dataset has passed our quality controls, testing gene expression correlations between cytokine ligand or receptor and target genes across many human bulk tissue datasets. The significance analysis function will identify differentially regulated target genes of the current cytokine. For each target gene, the module compares log fold change values from different studies with 0 through the two-sided Wilcoxon sand rank test. If the user applied row selectors, this module will only analyze the selected rows. The minimum count represents the threshold for the minimum number of studies required for a significant result. The p-value represents the threshold for statistical significance to include in the result table. The result table includes columns target gene ID, mean standard deviation of the log4 change across studies, and p-values. Clicking on the column will sort rows. For example, you can see BMP6 significantly downregulates CCL2 levels and may upregulate a few other genes across studies. Besides the statistical table, an interactive summary plot is available here. When you hover your mouse cursor over each dot, the plot will show the cell model and treatment conditions. The download button is available and can download the entire table in CSV format. The entire dataset of cytokine treatment is also available on the download page. The second usage of the search is querying cytokine regulators of a target gene. For example, you can input IL-8 and click search and get an interface like this. First, the system used the standard gene symbol for IL-8, which is CXL8. Second, we want to see upstream cytokine regulators of CXL8, but not its downstream targets. Therefore, we select the query as target genes. If you query a target gene that is not a cytokine, such as SOX4, you will only get one option of target genes. After clicking the Submit button, you will get a result table very similar to the one explained for the first usage of search, except the target gene column representing target genes of the cytokine will be replaced by the signal column representing cytokine signaling molecules regulating your target gene query. After clicking the Analysis button, you will get cytokines that differentially regulate your query gene. In the previous example of querying cytokines, the ID column represents target genes. Right now, it represents cytokine IDs. In the current analysis, the two-sided Wilcoxon sand rank test is comparing log4 change values of the same cytokine across different experiments against 0. An interactive plot is available together with the statistic table. Mouse hovering each dot will trigger the pop-out window of cell model and experimental conditions.